Okay, so are there any questions before we start today? So we finished up osmoregulation. So remember that osmoregulation, the primary organ that we have that's responsible for our osmoregulation would be what? What's our organ? Kidney, right? And what is the structural unit of the kidney that is responsible for filtration, reabsorption, and secretion? Well, the whole thing. What's the whole thing called? Nephron, right? And so the nephron is the functional unit that is in the kidney. And remember that the blood goes into the glomerulus where it's filtrated, and then through the tubules where it's modified before it goes into the collecting duct where it can be modified some more, because we talked about the role that ADH plays in retaining water or eliminating water, the lack thereof, right? And so that is going to be on the quiz on Wednesday, right? So that osmoregulation, so you want to make sure you review that. And you should read the chapter in your book on osmoregulation as well, okay? So today we're going to start on the nervous system. And so when we were talking about nervous systems, one of the things that we were talking about when we were talking about invertebrates is cephalization. So remember that cephalization is the brain, which is actually an aggregation of neurons at the anterior end of the organism. So that is your head end. So if we think about moving through, we're kind of weird in that we're bipedal. So like we move our whole body through, right? But if we were quadrupeds, if we were tetrapods, and we moved on all four legs, right? Our head would be the first thing that goes into the environment. And so we would be able to sense the environment and sense danger and possibly withdraw from that, right? So that is the significance of cephalization, and it correlates with bilateral symmetry. So if you'll remember, in the echinoderms, which include the sea stars, the larvae start out as bilaterally symmetric, and they have a brain, and they move through the environment in one direction, and then they settle down and become radially symmetric, and they lose their brain. So when we look at radially symmetric organisms, they tend to not have cephalization. So in your book, there's a little diagrams of the different types of nervous systems that we've seen as we've looked at the different organisms. There we go. Okay, so the cnidarian, what type of symmetry does the cnidarian have? It actually has radial symmetry, right? So it's radially symmetric. And so is the echinoderm. So you'll notice that they lack a brain. In the cnidarian, we have what is called a nerve net. So even like the sea anemones and the jellyfish do not have a brain, but they're able to have some sophisticated behavior even without the brain. And it just allows them to move through the environment in any direction. Okay. So in the planaria, we talked about planarian when we were doing orientation experiments, when we were looking at their movement towards or away from light. They have eye spots, which allow them to sense light, and then they have a brain, and then we have nerve cords, right? The one interesting thing about most of the invertebrates is that they have a nervous system that is ventral. So if we look at the honeybee, it has a brain, but it has a ventral nerve cord, and that is not what we see in the chordates. So remember, one of the characteristics of the chordates is that they have a hollow dorsal nerve cord. So dorsal means that it's in the back of our body. Hollow means that it's filled with cerebrospinal fluid. And then we have an aggregation of nerve cells at the anterior end, which would be the brain. Okay. Now, the mollusk, the octopus, has some really, really complicated behavior, and they're actually very, very intelligent. They're believed to be able to recognize individual humans and communicate with them. And they also have a brain, and then they have nerves that run out. Okay, but it's a different setup. It's not hollow. It's a solid nerve system. So it's not like what we see within the vertebrates. Yes.
Do octopus have big brains or little brains?
They have big brains.
They have that huge head.
Yep, and that's where their brain is located. And it's really interesting because they're thought to be super intelligent, like they can undo their cages and they can actually move, you know, some aquariums, they've actually caught them sneaking out at night and going to other terrariums and eating and then sneaking back in. So the weird thing about the octopus though is that it's kind of sad because they don't live very long. And so they have this complex behavior and then they might live 2 years and that is their lifespan. So they have a really sped up fast lifespan compared to vertebrates, for example.
You know, like the big ones, like they only live to be like 2 years?
Yes, yeah, and so there's a really wonderful book called The Soul of the Octopus, which talks about people and how they create these relationships with the octopus. And it's really sad because they have senility just like we do. And so when they get old, they get like they can't remember who you are, so they can't remember who their keepers are, and then they turn white. And so they tend to have kind of they described it as kind of Alzheimer's. They get kind of this weird behavior where they'll start acting really strangely right before they die. So anyway, so it's a very, very interesting nervous system. Okay, so let's look at our nervous system, and we're going to talk about initially the neurons, which are the individual cells that make up the nervous system. So these neurons are cells that are capable of communication. Via the release of neurotransmitters. So this is chemical communication, but we're going to also talk about the electrical communication, that the movement of charged particles down along the length of the cell, which then ultimately causes the release of neurotransmitters into the space between one neuron and the adjacent neuron. Okay. So when we look at neurons, we can say, I'm just going to draw my little neuron here. You don't have to draw it if you don't want to, but this is my little neuron, and it has these little branches, and then it has a long branch, and then there's a space between it and the adjacent neuron. So it kind of looks like this. Okay. So the neurons that have all the branches, the part of the neuron that has all the branches is called the cell body, and that's where the nucleus resides. And actually, that's where all the cellular machinery is that produces neurotransmitters. So if you think about, you know, it's kind of like a protein factory to produce neurotransmitters. And then we have these little branches, and these are called dendrites. So that's named after a tree. Like if you study trees, that's called dendrology, or branching is called dendrology, right? And so these are the many branches, and this is what receives, or where the neurotransmitter binds. So where the neurotransmitter binds. Okay. Now I've drawn really simple neurons, but I'll show you how they can actually be more complex. And sometimes it's really hard to tell what is the dendrite and what is the axon. And so this is my axon right here. And this transmits the signal in one direction. So we'll talk about what that signal is in a minute, or the impulse. So if I was to look at the signal, and I might use a different color here, the signal would be transmitted in this direction, down the length of the neuron. So information only flows in one direction. And so if you think about it, the motor neurons that go out to our muscles are arranged in one direction. And then, like if I step on a pin, the sensory neurons that would send that back up to my brain are oriented in the other direction. So we have like motor nerves and sensory nerves, we'll talk about in a minute. Okay. This right here is called the axonal terminal, or the end of the axon, and this releases neurotransmitters. This space between my neurons is called the synapse. Okay. Now, when we're looking at the orientation, we would say that this neuron right here is the presynaptic neuron. Right, so it's in front of the synapse. And this neuron right here would be the postsynaptic neuron. And that just kind of gives us some language to talk about what direction communication flows. So it flows from the presynaptic neuron to the postsynaptic neuron. Not neurons, but neuron. Sorry. Okay. Now the important thing to realize is that one neuron can actually synapse with hundreds or maybe even thousands of other neurons. So I've just drawn two here, but think about like if this neuron had synapses coming in, axons coming in and synapsing with it, it would get a lot of information. And then the neuron has to be able to take in all that information from the other neurons and decide whether or not to generate a stimulus or a signal or an action potential. Okay. So these are the neurons. And in your book, there's a diagram of the neuron. So you want to take a look at that diagram. Okay. So this is my diagram of the neuron from your book. And we're going to talk about these myelin sheaths and the role that the myelin sheaths play in a little bit, because that actually helps to speed up the conduction of the information down the length of the axon. So notice that the myelin sheath is only associated with the axon. And then these are the axonal terminals, and then that space is actually the synapse. So the synapse is the space between two neurons. So if we look at the different neurons, and I remember we looked at these neurons in the lab. So we looked at some slides, and you're like, what is all that stuff? And how do I tell which side is the dendrites and which side is the axon? It's really hard to tell just by looking at them. This one's pretty self-explanatory. This would be like a neuron that is sending information out to my muscles. So the cell body actually resides in my spinal cord, and then it sends it out to a muscle, and it's the axon that extends nice down and long. But this one actually has the cell body in the middle, right? And the dendrites and the axons, and the dendrites on one side, the axon would also be here. Okay, so just to show you that they're not always as simple as they draw them in the textbooks. Okay. So the other part of our nervous system includes glial cells. Okay, so these are helper cells. So some of the things that the glial cells do is that they help to maintain the interstitial fluid. So they maintain the fluid around the neurons, so interstitial fluid. Right? They clean up debris and fight off infection. And so for every one neuron, there's like 10 glial cells that are aiding in kind of creating the environment in which the neurons can survive. And so if we look at some examples of these from your book, I'm not going to make you memorize the different types, okay? So the astrocytes in particular are really important in creating the proper conditions, like the amounts of sugars and salts and ions that help to maintain the neuron. And then the microglial cells here actually are really important in cleaning up the debris. And then the only ones that you need to know specifically are the ones that create the myelin sheath. So I'm going to put create myelin sheath around the axon. So in the brain, in the central nervous system, which is abbreviated as CNS, this is my CNS, right? My central nervous system. These are called oligodendrocytes. And you've actually seen oligo before, because we talked about oligochaetes, right? We talked about earthworms are oligochaetes, right? They don't have many branches. So oligodendrocytes do not have many branches, but they form the sheath. And then what is it in the peripheral nervous system? Does anybody know? That creates the myelin sheath around the axon. In the it's named after a person. Does anybody know? These are called Schwann cells. Okay, and this is in the PNS. So this is in the PNS, which is the peripheral nervous system. Okay. So in your diagram here, they show Schwann cells wrapping around the axons in the peripheral nervous system. So this is in your book. And then the oligodendrocytes wrapping around the axons in your brain. Okay. So when we look at the brain and we look at the spinal cord, we can see that there's two different types of matter. So this relates the color. So if we look at the color, so this is a different topic, but if we look at the color of the nervous tissue. Okay. So we have what is called gray matter. Right? So the gray matter is where we have the nerve cell bodies and the dendrites. So this is primarily made up of nerve cell bodies. And then we have the white matter, which is made up of myelinated axons. Now, where the gray matter consists, or where it's located, sometimes we have, we do have two different names to describe if it's located in the central nervous system versus the peripheral nervous system. So in the gray matter, we have, so I'm just going to put here, I'm going to have nuclei. Okay, this is in the CNS. Okay, do not confuse that with the nucleus of a cell. So within, and I'll show you a picture of this in a minute, but within within the brain, we have little pieces of gray matter, and they look like maybe little nuts, right? And so they're called nuclei. And in the central nervous system, these are called ganglia. So aggregations of nerve cell bodies in the peripheral nervous system are called ganglia. If they're in the central nervous system, they're called nuclei. Okay. We can also talk about bundles of myelinated axons in the central nervous system versus the peripheral nervous system. Okay, so these would be called tracts in the central nervous system. So we can talk about the optic tract, right? And that would be the myelinated axons that are carrying stimuli from your eye to the cerebral cortex, where you are able to take that information and form a picture, right? So they're called tracts if they're in the CNS, and they're called nerves if they're in the PNS. So nerves are not the same thing as neurons. nerves, like your sciatic nerve that comes down your leg and innervates your whole leg and your foot, would be just bundles of myelinated axons. So it's quite amazing that these cells can be that long. And the nerves can be quite big, right? So they can be, you know, quite big in diameter, and they're just bundles of myelinated axons. Okay, so that's some terminology when we're trying to figure out what is going on in the central nervous system and the peripheral nervous system. Okay, so let's talk about neurophysiology. Oops. So the physiology is the mechanism, whereas we kind of just talked a little bit about anatomy and histology. So this is the mechanism. So how do the cells communicate, and what is that mechanism? So when we look at the physiology, we need to know, we need to define some terms, and we need to know what a membrane potential is. Okay. So the membrane potential is a difference in charge between the inside and the outside of a cell. So this is the difference in charge between the outside and the inside of the cell. So across the membrane. We can actually use little tiny voltmeters to measure that. So just like if you're trying to figure out if there's electricity running through a wire, you would use a voltmeter. If you're trying to figure out if there's electricity moving through a nerve or an axon, you would do the same thing. And they actually used giant axons from squid to determine this initially, right? So here's my little micro electrode, and squid and octopus have these giant neurons, and we can stick it in there, right? And then we can measure the difference between the inside and the outside. And so we record that using the voltage. Okay. So when a neuron is not doing anything, it has what is called a resting potential. So this is when there is no stimulus being transmitted. And the resting potential is generally about negative 70 millivolts, or MVs. And this is actually maintained using energy. And so the resting potential takes ATP to maintain. So the interesting thing about it is that even at rest, our nervous system is really energetically expensive to maintain. And so it makes sense that in animals that don't need a huge nervous system, they've tried to, you know, there's advantages to having a big nervous system, but there's definitely disadvantages to having a big nervous system because it takes so much energy. Yes?
Is that why the squid doesn't live so long?
Maybe, because maybe it's generally what happens, it's kind of interesting in mammals, the bigger you get, the longer you live. So if you think of an elephant versus a shrew, because the shrew actually has a much higher metabolic rate, and so they actually, if their heart pumps more and they're having to break down oxygen, so and so if the squid is having to use a lot of energy to maintain its nervous system, perhaps that is causing free radicals to do damage to the cells, which could lead to aging. Yes, but in general, the bigger you get, the longer. So it'd be interesting to see if that's the same thing with squid or octopus. Maybe if they're really big, they probably do live longer than the really small ones, I would imagine. But anyway, okay, so it takes ATP to maintain. And so if we look at the inside, so this is my axon, and this is my fluid outside the axon, right? So this is the outside, and this is my inside. Okay. So to show that it is negative, we would just put negative here, 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 right? And then we put positive on the outside. So it is negative 70 millivolts compared to the outside. Now, something else which is really important in the resting potential has to do with the ion concentrations. So we're going to put different ion concentrations, inside versus outside. So remember that ions are just charged atoms. And so if I look at the inside compared to the outside, I'm going to draw it in terms of like sodium and potassium. Sodium is really concentrated, and then I'm going to draw a little sodium, right? So what that means is sodium is more concentrated on the outside than on the inside. Okay. Conversely, I'm going to put a big K on the inside and a tiny little K on the outside. Okay, actually this is supposed to be smaller. Little K. Oops, this is supposed to be big. This one's supposed to be tiny. Sorry, doesn't look like that. Okay. So by diffusion, which way, just by physical force, would sodium tend to move? Inside, into the inside, or out to the outside? Inside, right? So sodium will tend to diffuse in. And so remember, diffusion is just like gravity. It just will happen no matter what. So sodium tends to diffuse in a little bit, and potassium will tend to diffuse out. Okay, but in order to set up this electrochemical gradient, is what they call it, we need them to be maintained as they are shown in our picture. So this is where we have the sodium-potassium pump. Okay, so this is huge, this idea. We have the sodium-potassium pump. Okay, so what this does is this counteracts diffusion. And so sodium is pumped out using energy. And potassium is pumped in. So we're setting up this difference in concentration gradients so that when we stimulate the neuron, we're actually going to open up some channels and cause the ions to flood in and flood out. Okay, but this is resting, so this is not when this is just at rest. Okay, so if we look at a diagram in your book, okay, this shows sodium on the outside, potassium on the inside. So notice that there's a lot more sodium on the outside than on the inside. There's just a few, right? And so this also shows that there's a channel here which is going to pump sodium out. So sodium is always being pumped out, and potassium is always being pumped in. And I know those are still all positive, so it kind of is confusing. Why is the inside negative when we're just changing the outside, or we're just changing two positive ions? But the answer here is that we have anions that are kind of stuck inside. Okay, so I'm gonna draw this here. So my anions, which are A negative. Okay. So if I draw A negative, what that means is that, this is kind of another idea, is that the anions are negatively charged particles that are stuck inside of the neuron. So they cannot leave. They're actually little amino acids. Okay? And so that's what keeps the inside more negatively charged than the outside, is that they cannot move. And anion is just the opposite of cation, right? So anion is just a negatively charged particle. Okay, so the next thing that we have to understand is what happens when we stimulate the neuron. And so we get what is called, we get what is called an action potential. So the action potential is a positive, and sometimes it's a positive 40 in your books, but this one has it just as a positive 30 millivolts. Okay, so this is what we call the impulse, right, or signal. And this positive charge moves like a wave down the length of the neuron. Right? We'll talk about how it actually jumps with myelinated neurons in a second. Okay, so what happens here is that we have gated channels. So we have what are called gated channels. These open when stimulated by a neurotransmitter, so they can open when they're stimulated by neurotransmitters, or by a change in voltage. So like a neurotransmitter could bind to a neuron and open up a gated channel so that the channel then allows sodium to flood in. Or the voltage, as it moves down like a wave, it will open up channels as the signal, as the action potential moves down the length of the neuron. Okay. So in the action potential, sodium channels open. So I'll put this as my events. This is the sequence of events. 1, sodium channels open. So my sodium floods in and it carries with it a positive charge. So we call this depolarization, because it goes from a negative 70 towards 0. So I'm going from a negative 70 millivolts and I'm moving towards 0 millivolts, right? So think that is becoming depolarized. So think about what polarization means, like extreme opposites. So it's moving from negative back to 0, so it's becoming depolarized, okay? So this is this first part right here. So here we have negative 70, right? And then it's moving towards 0, and that is what is called depolarization. So you need to know that, and I'll draw this so you can put it in your notes, but you need to know this graph, okay, that shows an action potential. Okay. The thing about this is that we have to reach a threshold, and this threshold is about a negative 55 millivolts, okay? Oops. So, okay. If threshold is met, then an action potential is produced. Okay. And an action potential is all or none. So we don't like have weak action potentials and strong action potentials. It's all or none. So once that threshold is met, the neuron will fire. And they always just fire at the same strength. So when we get like a bright light versus a dim light, it has to do with the number of action potentials that are being generated over time. So bright light will send lots of action potentials, and a dim light or a soft sensation would be slow action potentials. Okay, so that is the action potential is produced, and the third part is that the sodium channels close, potassium channels open, right? Potassium floods in. I forgot to, oh, excuse me, floods out. Oh, sorry, floods out. I forgot to put here that potassium moves in. I just didn't, I said it, but I didn't write it down. So I'll put sodium, potassium, excuse me, sodium floods in. Okay. I just forgot to write that down. Okay, potassium floods out. And so this carries the negative charge or the positive charge with it as it moves. And so the inside becomes re-negative. And so this is called repolarization. We're talking milliseconds. Yeah, so it's fast. You know, firing of action potential fast. Okay. Milliseconds. Okay, so repolarizes, and it generally overshoots the resting potential, and so we get what is called hyperpolarization. So hyper means more, right? So this is actually hyperpolarization is where we have a membrane potential that is more negative than negative 70 millivolts. So if it drops down low, then it'll be negative 70.
Does hyperpolarization always happen?
Yes. And it's the reason it kind of makes it so that the action potentials can't be so close together. So it kind of goes down and then it comes back up to resting. So it delays the initiation of another action potential. Okay, so if I'm going to, let's draw that little picture here for your notes. Just ignore that. Okay. So this is time in milliseconds. Okay. This is membrane potential. So this is voltage. Right. So we have resting, negative 70, depolarization, positive 30, hyperpolarization, and then back to resting. Okay. So I could write this down as this is my resting. And this would be a good diagram to have on the test, the quiz on Wednesday. This would be my threshold. So this is my threshold. Dotted line. This is depolarization. This is repolarization. And this is hyperpolarization. Okay, so those are the parts. This is an action potential. So you can label this graph action potential. So voltage increases. So voltage would be like be 0 here, right? Positive 30, negative, I know that's not to scale, but negative 70 would be down there. Okay, so that's what's going on. That is how a neuron is able to send an impulse. It's a wave of positive 30 millivolts down the length of the axon in particular. Okay, so this actually shows the thresholds where they're not met. So I want to point out that you can have your axon could be stimulated, but if it doesn't meet the threshold up here, they're saying like ignore that -10. But if it doesn't make the threshold, then it will not generate an action potential. Right? So these are just too weak to generate an action potential. This one finally makes the threshold, this one right here, and an action potential is generated. So the stimuli, the initial stimuli has to be strong enough to be carried to meet that threshold. Okay, so let's look at the conduction of the action potential. This depends upon the speed, specifically, depends upon the diameter of the axon. So it's kind of interesting. I would think this would be the opposite, but for some reason, the increase in the diameter means that there's an increase in the speed of transmission. So like crayfish that have that really fast reflex where they, it's a tail jerk reflex where they shoot away really fast. That is due to a giant axon that stimulates the muscles. It runs down their abdomen and into their muscles. Okay, so really fast actions demand giant axons. Same thing with squid and octopus have some really big axons. And we know that because the bigger the axon, the easier it is to work on when you're studying neurophysiology. Okay, the second thing is the presence of myelination. Okay. So if you think about it, once an action potential has been generated and a threshold has been met, what happens is that it just travels like a wave down the axon. And so this is a diagram from your book that shows the positive depolarized part and how it moves, right? So it's moving down the length. And if it's not, this is not myelinated, it's going to take a lot longer for it to move, right? So an unmyelinated skinny axon is going to be really slow, right? So if we look at what happens with myelination, okay, you could draw this in your book. So this is my axon. And what I have here are the Schwann cells or the oligodendrocytes, depending upon where we are in the nervous system. Okay, so this is the myelination. Okay, so the cells are kind of interesting because they actually have a nucleus and they actually wrap around the axon and they insulate it. So it's like putting electrical insulation, you know, that black sticky tape around your wires to protect yourself from getting shocked, right? But you insulate it so that the electricity isn't lost. And so what happens is that the action potential travels down the whole length, but it only has to open up channels, gated channels, in what are called the nodes of Ranvier. So I'm going to put, this is my node of Ranvier. This is my, in this case, the Schwann cell. Okay. And so notice that it jumps, and this jumping of the action potential down the length is called saltatory conduction. So saltation is jumping. So you could describe a kangaroo as having saltatory locomotion. So saltatory conduction means that the action potential jumps from one node to the other. And so it is much faster, okay? So saltatory conduction is much faster. So it's kind of interesting, when babies are born, sometimes you'll notice that like when they first start to move, their movements will be really jerky. Like or they might be slow and uncoordinated. So when babies are born, a lot of their axons are unmyelinated, and then as they use them, the axons become more myelinated and the movements become more fluid. So does anybody know what autoimmune disease attacks the myelin sheath and demyelinates the neurons?
Parkinson's or something?
Not Parkinson's, MS. So multiple sclerosis. Or MS is an autoimmune disease where the immune system, for some reason, attacks the Schwann cells. And so this is said to be a demyelinating disease. Parkinson's is actually due to the death of neurons in a part of your brain called I think it's called the substantia nigra. It's midbrain. But this is multiple sclerosis is actually demyelinating. So the interesting thing about it is it does not damage the neurons. So really all the symptoms that you see from multiple sclerosis, the slowing down, the loss of neuromuscular coordination, is due to losing the myelin. So that's kind of how we know how important myelin is. There's another disease which is kind of similar to this, but it is tends to cure itself, and this is called Guillain-Barré. And this tends to repair itself slowly. So instead of constantly attacking the myelin, it's like, I've known somebody that had this. She was actually an instructor here, and she had to leave her job. She was a PE instructor. And she got a really bad viral infection, and then she got, she was just getting over it, and then all of a sudden she woke up and she was paralyzed. And what happens is they take you to the hospital, and then over a period of like a year, your body slowly starts to repair the damage that your immune system has done. And so the myelin starts to be rebuilt around the neurons, but it takes a long time, right? So she would have to learn how to re-walk, for example, because of the Guillain-Barré syndrome. Yeah.
You ever heard of people using D-venom treatment for MS?
No.
I've heard about it. I've seen it. I've known people that do it, and they say it really helps.
Interesting. So sometimes people have, they get better a little bit, and that's because the immune system stops attacking the myelin, and the myelin rebuilds itself, but then the immune system will re-attack it. And so you have these periods of relapse where you start to feel better. The interesting, I did mention that they use hookworms sometimes to treat MS, not in the United States, but elsewhere, okay? Because that helps to give the immune system something to attack rather than attacking the Schwann cells. Okay, and I've actually known dogs that had Guillain-Barré, and generally they have to put them down, right? Because if you can imagine like having a lab with this, spending a year in rehab would just be too expensive. You can't rehab your dog for a year, and then not able to move, etc. Okay, so myelination also plays a role in learning. And so I'm going to show you this video that talks about how best to learn, and they refer to this idea of myelination in this video. Oops.
Eggs get fertilized, and by that I mean sperm.
Want that. Okay, where did it go?
Mastering any physical skill, be it performing a pirouette, playing an instrument, or throwing a baseball, takes practice. Practice is the repetition of an action with the goal of improvement, and it helps us perform with more ease, speed, and confidence. So what does practice do in our brains to make us better at things? Our brains have two kinds of neural tissue, gray matter and white matter. The gray matter processes information in the brain, directing signals and sensory stimuli to nerve cells, while white matter is mostly made up of fatty tissue and nerve fibers. In order for our bodies to move, information needs to travel from the brain's gray matter down the spinal cord through a chain of nerve fibers called axons to our muscles. So how does practice or repetition affect the inner workings of our brains? The axons that exist in the white matter are wrapped with a fatty substance called myelin, and it's this myelin covering or sheath that seems to change with practice. Myelin is similar to insulation on electrical cables. It prevents energy loss from electrical signals that the brain uses, moving them more efficiently along neural pathways. Some recent studies in mice suggest that the repetition of a physical motion increases the layers of myelin sheath that insulates the axons. And the more layers, the greater the insulation around the axon chains, forming a sort of superhighway for information connecting your brain to your muscles. So while many athletes and performers attribute their successes to muscle memory, muscles themselves don't really have memory. Rather, it may be the myelination of neural pathways that give these athletes and performers their edge with faster and more efficient neural pathways. There are many theories that attempt to quantify the number of hours, days, and even years of practice that it takes to master a skill. While we don't yet have a magic number, we do know that mastery isn't simply about the amount of hours of practice. It's also the quality and effectiveness of that practice. Effective practice is consistent, intensely focused, and targets content or weaknesses that lie at the edge of one's current abilities. So if effective practice is the key, how can we get the most out of our practice time? Try these tips. Focus on the task at hand. Minimize potential distractions by turning off the computer or TV and putting your cell phone on airplane mode. In one study, researchers observed 260 students studying. On average, those students were able to stay on task for only 6 minutes at a time. Laptops, smartphones, and particularly Facebook were the root of most distractions. Start out slowly or in slow motion. Coordination is built with repetitions, whether correct or incorrect. If you gradually increase the speed of the quality repetitions, you have a better chance of doing them correctly. Next, frequent repetitions with allotted breaks are common practice habits of elite performers. Studies have shown that many top athletes, musicians, and dancers spend 50 to 60 hours per week on activities related to their craft. Many divide their time used for effective practice into multiple daily practice sessions of limited duration. And finally, practice in your brain in vivid detail. It's a bit surprising, but a number of studies suggest that once a physical motion has been established, it can be reinforced just by imagining it. In one study, 144 basketball players were divided into 2 groups. Group A physically practiced one-handed free throws, while Group B only mentally practiced them. When they were tested at the end of the 2-week experiment, the intermediate and experienced players in both groups had improved by nearly the same amount. As scientists get closer to unraveling the secrets of our brains, our understanding of effective practice will only improve. In the meantime, effective practice is the best way we have of pushing our individual limits, achieving new heights, and maximizing our potential.
Okay, so that's what they say, study every day, right? Okay. Any questions about that? Okay, so let's take a little break. We'll take a 5-minute break, and then we'll come back and talk a little bit more about neurophysiology, and then we'll talk about structure. Oh, and if you want to, this is the human brain. So this is half, so you can come over here and get an idea. You can see the difference between the gray matter and the white matter. That is a person with a light. And now it's your job now to the job. facilitator.
It's definitely George.
This animation shows brain development from conception through birth.
Underneath, with the newfangled
First 4
cortex,
weeks after.
the brain stem, the limbic system, and the basal ganglia date back to the mastodons.
Okay. So we talked about what the action potential is, what the impulse is, how it moves down the length of the neuron in one direction. But I want to talk about how the communication across the synapse occurs. And I'm going to specifically talk about the chemical synapse. So there is an electrical synapse in some organs, which just simply create channels through which ions flow. But the chemical synapse is where we have the diffusion of neurotransmitters across the space. Okay, so diffusion just means they go from a higher concentration to a lower concentration. The space is really small, so diffusion can happen quite quickly. But what we have to do is we have to turn the electrical impulse into a chemical response, right? So the chemicals have to respond. And this is how we communicate across the synapse. So the first thing that has to happen is I'll just put AP, that's action potential, okay, reaches the end of the axon. The second thing that has to happen is calcium channels open and allow calcium to flood in. So this is an ion we haven't talked about yet in terms of its significance, but calcium, and calcium has 2 positive charges, so it's generally like a 2+ or +2 usually. Okay, so calcium ion channels open, and calcium floods in. So calcium is generally pretty low, so it just opens and the calcium moves from a higher concentration to a lower concentration. So what that does is it causes synaptic vesicles to fuse. So synaptic vesicles, which are filled with neurotransmitters, fuse to outer membrane and release neurotransmitters. Calcium is really interesting because its levels outside the neurons are obviously really highly regulated by those glial cells. And so if you have too much calcium or too little calcium, that can directly affect your nervous system. It can actually kill you if you have too much or too low. So calcium ion concentrations are regulated in the nervous system. Okay, so then the neurotransmitters cross across the synapse and will bind to receptors on the postsynaptic neuron. So they diffuse across synapse and they bind to receptors on the postsynaptic neuron. And that would be the dendrites, right? So that would be the dendrites, not the axon. So the neurotransmitters diffuse across the synapse. Okay, so some neurotransmitters are excitatory and some are inhibitory. So if it is excitatory, so if I have an excitatory neurotransmitter, what this causes is sodium channels open, right? And depolarization occurs. So an example of this would be like dopamine. Right? So dopamine is excitatory. It increases the number of action potentials produced. And there are some drugs like cocaine that increase the amount of dopamine in the synapse, and so you get a rush. Because with cocaine in the system, dopamine is more present and more action potentials are produced. Okay, so that's excitatory. We can also talk about inhibitory. And so one that's inhibitory in our body, in the central nervous system, is serotonin. Okay, so serotonin is the example. And in this case, chloride ion channels open. We haven't talked about chloride either. Okay, and what this causes is it causes hyperpolarization. So it moves the neuron away from the threshold. So it decreases the likelihood that an action potential will be produced. Okay, so it makes the inside even more negatively charged than resting. Okay, So the question is we have lots of drugs that are called SRIs. So these are commonly prescribed by doctors. Okay, and an SRI stands for serotonin reuptake inhibitor.
Are they SSRIs?
Serotonin, no, SRIs. SS, two S's. I'm not sure. I think they're just SRIs, but I'm not sure. I haven't seen the SSRI. I haven't seen that. I look it up, but you can Google it and see. Okay, so serotonin, that's the serotonin reuptake inhibitor. So what this means is that this keeps the serotonin in the synapse longer than it normally would be. And so this decreases the number of action potentials that would be produced. And so they oftentimes give this for depression, which is kind of weird, because if you think about depression, you think depressed nervous system, right? But really, when people are outwardly depressed, sometimes they are experiencing a lot of anxiety, right? So if you've ever experienced being so anxious that your mind is going like crazy, you're worried about all these different things, and you're so anxious that but when people look at you and they say you're depressed, right? You can't get off the couch. But what happens is that you're so anxious that you can't do anything. So if you increase your serotonin levels in your brain, then you tend to be less anxious and you tend to be more able to do stuff. Serotonin also affects your skeletal muscles. We won't go into that, but so when you take SRIs, oftentimes you have to get up and move because your skeletal muscles kind of like want to move, and so it's hard to not do stuff because you have to get up and move. Did you look it up?
It's an SSRI, selective serotonin.
Oh, selective. Okay. Yeah. Selective serotonin reuptake inhibitor. Okay. Okay. So those are the different types of neurotransmitters. So let's look at the diagram in your book. So this would also be a good diagram to be able to label. So notice that they say that this is the presynaptic neuron. So this is the action potentials are being passed down the length of this. This is my synapse, so this is my postsynaptic neuron. Notice calcium channels open and calcium moves in. These are my synaptic vesicles. So these are filled with neurotransmitters, and when calcium is present, what happens is it causes these vesicles to fuse to the outer membrane, and then these would be the neurotransmitters. So the neurotransmitters come across, right? They bind to chemically gated channels, and they can cause those channels to open. And so if this is an excitatory neurotransmitter, it would cause sodium ion channels to open. And if enough sodium ion channels open that it reaches the threshold, then this postsynaptic neuron will carry the impulse on. If not enough are open, then, or in some, remember we could have hundreds of synapses. Sometimes you get excitatory, I'm sure every time, you get excitatory and inhibitory being passed to the same neuron, and the neuron has to integrate that information. It has to say, you know, kind of decides, well, this, that's anthropomorphic, but excitatory is greater than the inhibitory, and it reaches the threshold and action potential will be generated. Okay, so this shows the reuptake, right? So like Prozac and Zoloft and those kinds of things inhibit the reuptake. Some, like I mentioned cocaine prevents the enzymatic degradation of dopamine. And so you have more dopamine in the synapse. Okay. So this would actually show a excitatory action potential. Okay. So we're going to watch a little video that talks about neurons and how they work. To just kind of summarize, this is just like a 5-minute video. It summarizes some of the things we've been talking about.
Underneath the newfangled cortex, the brainstem, the limbic system, and the basal ganglia, date back to the mastodons, the dinosaurs, and the first amphibians. In humans, the old parts of the brain oversee emotions and help build memories. They control heart rate and breathing. They also form intimate connections with the new brain, the cortex. Though less than a quarter-inch thick, the cortex is the brain's crowning glory. Among its roles, the cortex is our reality check. It filters and orders the outside world for us and allows us to see, touch, hear, and speak. The cortex is also the human thinking cap. All our plans, thoughts, and ideas originate in this layer. The cortex is packed with nerve cells. About 2/3 of all our neurons operate here. A piece of cortex tissue, no larger than a pinhead, can house 30,000 of these cells. Each neuron has a job: to communicate with other neurons. The brain works by forming networks among these cells. The long, spiny branches of the networks create a neural forest of astounding intricacy. Neurons use these communication lines to talk to each other with electric and chemical signals. Here under microscope are two neurons linking up. Though it may look like they fuse together, neurons don't actually touch each other. A closer look reveals that a tiny gap called the synapse separates their branches. This is where a message passes from one neuron to the next. The message comes from here, small sacs that store chemical molecules. When stimulated, these sacs release their molecules which cross the cell membrane into the synaptic gap. An electric zap allows this to happen. Meantime, the receiving neuron has special welcome sites for the incoming molecules. These receptor sites bind with the molecules. When they do, special gates open up. The gates let in a flood of charged particles, sodium and potassium ions, which start up a new electrical signal in the receiving neuron. This simple chain of events, an electrical zap, followed by chemical changes, followed by another electrical zap, is the basis of all brain activity. It's how neurons speak to each other. Here, then, is the key to the brain's complexity. There are 100 billion neurons in the brain. Each neuron, like a bit player in a grand production, processes its information, then hooks up with as many as 50,000 other neurons to send and receive messages. 100 billion neurons times 50,000 connections. It's this complexity that allows us to think imaginatively. On their own, neurons aren't very bright, but put 100 billion of them together in a small space and let them all start talking to each other, and you start to get brainstorming. The trillions of neural networks, like an improvisational orchestra, create new ideas and connect different thoughts in a whimsical and sometimes inspired fashion. It's this impromptu ability to produce new things in our brains that allows us to progress, that quite simply takes us from here to here.
Okay, so that shows the complexity of the interactions between neurons and synaptogenesis. So when we talk about learning, the learning would involve creating new connections, which is sometimes referred to as synaptogenesis. We'll talk more about that in a second. Okay. Skip that, sorry. Okay, so we're going to talk about the central nervous system today, and the central nervous system consists of two parts. This is the brain, which is protected by the skull, and then our spinal cord, which is connected by or protected by the vertebrae, right? So in vertebrates, we have both the cranium and the spinal column, right? A vertebrae that protects the spinal cord. When we look at the brain in particular, we have what is called a forebrain, a midbrain, and a hindbrain. And when we look at our brain, it's kind of weird. It's like, I don't understand where those terms come from, but it actually comes from the development. So if we think about our nervous system early on in embryonic development, it actually looks like this, right? So this would be the anterior end. So that's like the forebrain, right? And what happens in us is that that anterior end gets really big and it actually folds back on itself, right? And it becomes like the 80% of our brain, which is what is referred to as the cerebrum, okay? So the cerebrum in us has gotten much bigger, okay? So this is the cerebrum. So we have two right and left, and so these are called the cerebral hemispheres. So this is composed of two cerebral hemispheres. Okay. Now when we look at the cerebrum, as was shown in that video, the outside is called the cortex. So we have the outer cortex. And what color was it? It was gray. It's actually kind of brown in that picture, and actually it's kind of a tan, but it's gray. And so what does it contain? Does it contain axons or cell bodies? Axons, right? Because the axons are the insulation. They're like lipids, right? And so these are the cell bodies. So it's kind of weird. The cell bodies are on the outside of our cerebral hemisphere. And this is very thin. So this is thin, right? This is what is responsible for conscious thought. So our cerebral cortex is the only part of the brain that is conscious. Right. And so when people say you're brain dead, that means that when they put electrodes on your skull, they don't see any electrical activity in the cerebral cortex, and so that cannot be stimulated to get back. So once you lose the electrical activity in your cortex, even if the rest of your brain is working, you are technically brain dead, right? So you can swallow and you can blink and you can move your eyes, but if the cerebral cortex isn't working, then you're not conscious, right? And so even when we're unconscious, we have some electrical activity. It's only when we're brain dead that we lose the electrical activity in the cerebral cortex. Okay, so the inner part is white matter. Right, and so this is white matter, and it consists of myelinated axons. And in the central nervous system are those tracts or nerves. You remember? Are there nerves in your brain or are there tracts in your brain? Tracts. Okay. So if we look at our cerebrum, one of the interesting things about mammals in particular is that their cerebrum, the outermost part, is folded. And we've talked about this before, but what this does is this increases the surface area of the cortex, right? So these parts that stick up, these ridges, are called gyri. So the gyri, G-Y-R-I, these are ridges in our cerebral cortex. And then the valleys, or the indentation, are called sulci, right? So these are indentations. Right? So what this means is that we have a cerebral cortex that has lots of surface area. So you can see on this diagram, see where all those little grooves and ridges are. Those are the gyri and the sulci of the cerebral hemisphere. So there's just half a brain here, so there's only one hemisphere in the preserved human brain there. Okay, so if we compare this to other animals, okay, so if you compare it, oh, I don't have the, so if you looked at a fish brain, or if you looked, even here, the rat brain, but if you looked at other, besides mammals, so this just shows the mammals' brains, they, like if you look at the brain of a reptile, of a brain of a fish or a frog, they would not have these ridges and grooves, right? But we see in mammals, and I think even in the rat, you have a few ridges and grooves. You can see that they have a very large cerebrum. So that's the largest part of our brain, and that is part of the forebrain. Okay. Now, if we look at the conscious part of that brain, okay, this being the cerebral cortex, right? The two hemispheres are connected to one another by a band of white matter, which is called the corpus callosum. So I want you to write down the corpus callosum is white matter that connects the two hemispheres. So information can cross the cerebral or the corpus callosum. And so it's kind of interesting the way that our brain works, but information that is coming from the right side of the body, actually eventually, it could cross at the spinal cord too, but eventually it's going to cross over and it's going to light up the opposite side of your brain. So if I touch my right foot, the action potential is going to go to my left hemisphere. Okay, and so you can see the ridges and the grooves, and this is the separation. This is actually a fluid-filled space because we have a hollow dorsal nervous system. So our brain has holes in it, or spaces that are filled with cerebral spinal fluid, and those spaces are called ventricles. So if we look at the, a little ahead of myself. Oh, here's just another picture. So this outer part would be the gray matter, and it would be just the outermost part of this is where the cell bodies reside, and that's the conscious part of the brain. And then the inner part, including the corpus callosum in this section, so this would be like heading your head down coronal section wise. You can see the corpus callosum, and then you can see the ventricles, which would be filled with fluid. So this is a picture right out of your book. Okay, so if we look at the cerebral cortex, we can see that different parts of the cerebral cortex are responsible for sensing, consciously sensing different information. Okay, so we can say that we have a map. Let's put map of cortex. Right, and so the way that they created this map is that they would give a person sensory information like sound, right? And then they would look to see where in the cortex where the action potential is generated, right? So we have what is called the auditory cortex. right? So this would be responsible for consciously perceiving sound. We have also the olfactory cortex. Right? This would be smell. We have the visual cortex, and this would be images. Okay. Does anybody know what the taste is called? It's got a weird name. It starts with a G. It's called the gustatory. And so this is taste. Okay. So if I'm smelling something, it should go to the part of my brain, which would be like right here near the temporal lobe, parietal lobe area. If I'm smelling something, I should send a signal there. There's some people that get sensations crossed, and this is what is called synesthesia. And sometimes it's associated with synesthesia, autism, right? But it can also be kind of associated with being brilliant at something. So this is the mixing of the senses. Yes. So you smell something and you associate a smell with a sound. Or it could be that you look at a number and you see a color.
So if you have that, you're more inclined to use.
Yes. And the reason is that you think of it differently. So if I can see sounds, if like a sound is a certain color, then I can create a really interesting composition, and it tends to be completely different because I create a composition not just based upon sound, but also the way the colors
So
mix.
does that work with like, is it beneficiary because the memory is better because it's mapped out that way? Or?
No, it just allows, it seems to be in the past, people that have made huge leaps and bounds in whatever field they're studying tend to have this. So like if I'm a mathematician and each number is associated with color or a sound or whatever, then it kind of takes your mathematics to a higher level, right? But it can also be very distracting and confusing if you're, every time you hear something, you taste something, that can kind of freak you out. And so with autism, those people tend to be overstimulated because of the sensory mixing.
Well, this is something that happens when people take LSD.
Yes. Oh yeah, so this mixing of senses. And some people claim that that gives them brilliant insights as well. Okay, so somatosensory cortex. So the last thing we're going to talk about is the somatosensory and the motor cortex. So what do you think soma means? What is soma? Body, right? So these are the body senses. So these tend to be skin sensations. Okay. Somatosensory, skin sensations. But they're also associated with joints as well. So I have signals coming from my shoulder joint telling me if my arm is straight out or if it's down by my side, right? So those are called proprioceptors, right? So I can tell where my hand is relative to the rest of my body, right? So that's somatosensory. We also have the motor cortex. So motor in this term means locomotion, it means movement. So these contain motor neurons that send signals to muscles. So these are sensory neurons that receive signals. These send signals. And it's very interesting, the map that they create And when I was learning this, 20 some years ago, they thought that once you became an adult, your cortex was set in stone, like you had no plasticity. And now there's this whole new idea, and it was like this huge upheaval, and it probably still is that our minds can actually change, and we're actually have a lot more plasticity than we thought. And so if we look at a diagram that shows the difference between the motor and the sensory cortex, I'll show you what I mean, okay? So these are, you don't need to know where these are located, but they're located, and this is just a cross section. So the motor is in front of the somatosensory, okay? So the cool thing about this is this shows a person, and then it shows what proportion of your cortex is actually associated with that body part. So you notice the eyes and nose, this would be touch, right? So we have a lot of our somatosensory cortex associated with this part of the body. But if you looked at like your upper arm or your elbow or your hip, right, it's less, right? So it was thought that this was not changeable, right? But what they've discovered now is, let's say that you amputate an arm, right? Or you lose, in this case, it would be your visual cortex, if you lose sight, right? But let's say you amputated this arm, right? Then there can be some problems. You can have what is called phantom limb syndrome. And what phantom limb syndrome is the person still feels their appendage even though it's not there. And that is not necessarily a problem unless their appendage is in pain. So sometimes they feel like their arm is on fire, right? So if you were walking around, you didn't have an arm, but it felt like it was on fire, how distracting that would be, right? So the idea here is that this part of the somatosensory cortex needs to do something. And so it can sometimes create phantom limbs, but if it doesn't create a phantom limb, it is going to be incorporated into another part of the body. So let's say maybe I lose my lower half, maybe that part now becomes associated with my shoulder, and I'm more sensitive in my shoulder than I was previously. And so there's this idea that if you use a body part, let's say you decide you're going to be able to move your toes in a complicated way, maybe to grasp or to pick something up, what's going to happen is that if you use your toes more, then more parts of your somatosensory and your motor cortex are going to be associated with your toes. So let's say you didn't have arms and you did everything with your feet, right? Then a part of your that cortex would become associated with that. Okay, so there's lots of new books that talk about how you can change your brain by just simply changing the environment and what you do with it. Okay, so remember that this cortex is just the conscious. This is the only part of our brain that is thought to be conscious. And you'll notice that other organisms have it. This is the crazy thing. When I was in school, they said humans are the only creatures with consciousness. It's like, really? Seriously? So they've kind of finally, it was just like 2 years ago that scientists wrote this manifesto that says, we as scientists think that other animals have consciousness, you know? So like your cat probably knows that it is different from, you know, you and other things in its environment, right? So I believe that other animals other than humans have consciousness, especially if, you know, if we look at their cortex.
Is that how they figured out like that fish can feel pain and things like that?
Yes. So if you stimulate them, like, so if you prick your thumb, right, they can put sensors on your head, and this part of your brain will fire. It will light up because the action potential is sent to there, right? So sometimes you can have sensory mixing here. So like if I touch my foot and I feel it in my hand, that means that the action potential went to the wrong part of my cerebral cortex, right? So the only difference between, it's kind of like you're a puppet, you know, because really the only difference between sensing your thumb and your pinky is what's happening in your brain, right? So where that action potential is going, it's the same action potential. It's all or none action potential is sent in. Okay, so we'll stop there for today, but remember that you have a quiz on Wednesday over osmoregulation and nervous system. Remember, practice reinforces myelination in your brain. So if you study a little bit every day, specifically right before you go to bed, they've discovered that if you study right before you go to bed, you're more likely to put that information in long-term memory.
That's why I procrastinate. So I mentioned that, like,
If it's something that I have to learn, I will lay in bed and I will visualize it.
Yeah, I do the same thing.
And that is how and so that visualization.
I'll be up late studying and doing stuff ready for bed. Like I'll stay up late.
We could take a nap. You could
Yeah.
study and then take a nap.
Does that
Yeah.
help too?
Yeah. Yeah.
you know, like that.
Excuse from taking a nap.
Just until the next day.